and I'm extremely excited to be here today to talk with everybody. So let's get to it. I'm going to share my screen. Okay, so first I have a disclosure. I have two disclosures, actually. Some of the eye tracking tests that I'll be talking with you about today are licensed through the University of California. And if someone uses them for profit, I will get a small royalty, but they're always free for research use. The second disclosure is that I talk really fast, and I'm trying hard not to, but I just wanted to mention that. Okay, so I'm the co-director of the Autism Center here at UCSD, and we do a lot of different things to try to understand autism from the first stages of development. We do early screening and diagnosis and prevalence, genetics, brain imaging, stem cell, eye tracking research that I'll be focusing a lot on today. We've got over 20 scientists and staff, and it is co-directed by myself and Dr. Eric Crochane. And as Dominique mentioned, we really want to get our findings out into the public as quickly as possible. So we publish around 20 papers per year. And I want to start this lecture by honoring individuals with autism. Here on the screen, you can see three amazing individuals, and despite the challenges that they must have faced, achieved at least some of their goals and dreams. We have Stephen Wiltshire, who's an artist, Temple Grandin, who's an author and a scientist, and Ayush Bambadi, who is an illustrator. And unfortunately, though, these individuals are more of the exception rather than the rule. We know from research from the CDC and lots of other studies that individuals with autism still have struggles. 38% of individuals have intellectual disability. 61% have IQs below 85. But for me, the biggest motivator and why I got into this field a long, long time ago, actually when I was 19 as an undergraduate, the average age of first diagnosis on average across America hovers around age 4 years. And that is kind of my call to action. I'd like to change that. And the reason is because we know that plasticity is much higher during the early ages, although you can actually change, and a lot of plasticity can happen throughout the lifespan, but is definitely highest during the first years of life. And there's research that suggests that autism begins during prenatal life. It begins in the womb. Well, how do we know that? There's multiple studies, but I'm just going to highlight two that was conducted by our center. One was led by Eric Crochane, published in JAMA. Basically, what that study did was they looked at tissue from individuals with autism who had passed away, and there were stereologists blind to diagnosis who just counted the number of brain cells. And what you can see on this image here is that individuals with autism here are in circles, and individuals who are neurotypical are shown in triangle. And here, this is the total neuron count in dorsolateral prefrontal cortex. You can see right here on this plot, this little line here shows the median value. You can see individuals with autism have way more brain cells, particularly in frontal cortex, than non-ASD individuals, 67% more brain cells. And because your brains are developed and all the neurons you're ever going to have, for the most part, occur during pregnancy, the idea is, well, if individuals with autism have this enormous number of brain cells, this is probably something that happened during pregnancy because that's when brain cells are made. Likewise, we have another study that was published in the New England Journal of Medicine led by Rich Stoner. The brain actually has 6 layers, right? We have the cortex has 6 layers, and you can use different stains to kind of check and see what that layering looks like. And here's just a close-up view of the layering of an individual on the spectrum. You can see there are some places where the layering isn't as pronounced. And so again, this is evidence that because this cortex is built during prenatal life, that whatever's going on with autism is happening during pregnancy. It's not a postnatal event for the most part, like vaccines or something like that. It's going on early. And there are lots of papers that are thinking about when autism begins. And if you do a search on PubMed, there are 3,082 results that will pop up if you type in autism and prenatal. So it's a hot topic right now. And if you think about it from the perspective of how the brain develops, you can kind of see why I feel like there's an urgency to understand kids who could use some extra support as early as possible. This is a slide. It's super old, from Connell in 1939, but I really like it because it shows what brain cells look like when a baby's born. You can see a newborn, it has some cells, but they're tiny. There's not a lot of connections. By 1 month, a little bigger. By 6 months, you can see some local circuitry that's starting to form, and these are frontal cortex cells. And by 2 years, you can see the neurons are all talking to each other. But when does the average child with autism first get identified and start receiving their services? Around age 3 to 4. And I'm thinking that, well, what if we could identify kids, let's say at 12 months as standard of care, get them the support and services they need, because when the brain is starting to form and all these connections are forming, we can optimize connections that might support language, might support social engagement. It's a lot easier to do it while it's happening than after the fact. And so that's a lot of the motivation for why the research that I do. And there are some compelling studies that so treatment started by 18 months can really result in significant gains, not just in the field of autism, but in other fields. Like, for example, babies that are born deaf that get cochlear and hearing, these like cochlear implants to help with their hearing. In this particular graph that I'm going to show you, babies that got their cochlear implants before 18 months are shown as yellow circles. Babies that got them after 18 months are shown as blue diamonds or blue triangles. And babies that never got any cochlear implants are shown as green squares. You can see here on this particular figure by Sejas, you can see that we're looking at language, math, reading, writing, and science scores for these three individuals, three group types. And you can see these orange dots are pretty much the highest across the board. And these are kids that got their intervention before 18 months, and everybody else either didn't get the intervention or got it after 18 months. So you can see that in this population, 18 months is a really critical age. There's also a study that I really like, published by Chuck Nelson's group, published in Science a long time ago. They looked at orphans who were basically infants that were abandoned and placed in an orphanage in Romania. Doesn't have a lot of stimulation, a lot of interventions or support going on there. And some of the babies got out and were placed into foster care at different ages, but some never got to leave. And you can see here on this little table, looking at the age of placement, babies that got out of the orphanage and put into foster care with a more typical stimulating environment at 2.5, so 30 months, their IQs or developmental quotients were pretty much was 80. But the babies that got out and got into a more nurturing environment between 0 and 18 months was 95. So that's a 15-point difference. And here we move now to a recent study of autism published by Guthrie and colleagues. Basically, children who got their interventions started by 18 months had receptive language scores that were 10 points higher than individuals that got their treatment starting a little bit later that were only 3 points higher post-treatment. So early seems to, be really important. And so for me, I didn't really have time to start with the whole story, but I started working with individuals with autism when I was a teenager, and mainly older individuals who didn't receive a lot of intervention and they were just having a lot of struggles. And so I was pretty adamant that I wanted to do something where I was going to find kids as young as possible and try to help out. But if autism isn't usually identified until around age 3 or 4, how am I going to discover early biomarkers and try to support kids at super young ages like 1? I wanted the first birthday. So to do this, I came up with this idea of the Get Set Early model, where I mobilized a network of over 240 pediatricians here in San Diego County to screen for autism and other conditions starting at the 12-month well baby checkup. And they all operate in a standardized way. And anybody that kind of fails this parent report screen, the CSPS that we use, comes to our center. And then we come to our center, we do a workup, we do eye tracking, take a blood sample, and we refer for services on the very same day if a child's not meeting any milestone, any social, language, cognitive. And in the Get Set Early model, S stands for screen, E stands for evaluate, T stands for treat. As I said, we use Amy Weatherby's CSPS. It's super fast. It's only 24 questions. But what I like about it is it detects any delay, not just autism. And, the label isn't really that important for me. It's just finding somebody who's just not quite on the right track. And let's just go ahead and provide some services to help them kind of achieve age expected milestones. And kids who are developmental delayed or language delayed, as well as kids on the spectrum, will fail the CSPS. They get referred to our center. 25% all false positives. And so these are some of the pediatricians who have supported this project in San Diego. And we've screened over 150,000 babies to date, 40,000 more in Phoenix, with Chris Smith, who is the site PI there. So just quickly, when you do the Get Set Early model, this particular study was with 57,000 kids screened. The modal age at screen was 12 months. And children who got screened at 12 months, you can see here in this first bar graph, the average age of first diagnosis was around 15 months, because it takes a little bit of time to get in and get your evaluation. And then here we refer for immediate treatment the same day, and children in our study received around 10 hours of intervention. And we're currently following up the kids at school age to see if this super early intervention, what kind of impact that had. So this is kind of the backdrop, because when I'm showing you data in the rest of this presentation about eye tracking and about brain imaging and a little genetics, it's a lot of the kids are super young. So, this is the method that we received super young toddlers to participate in our studies. It's really the only way that you can do it. Unlike you can do a baby sibling design, but then you're only studying multiplex families. So it's kind of, you do this general population screen starting at 12 months, or you can do a baby sibling design. And because we're going to be talking a lot about biomarkers, there are certain metrics that scientists use to understand kind of the quality of the biomarkers or the accuracy of the biomarkers, right? So let's say you're screening a baby, and you want to see if any marker that you're finding is accurate or not. So baby, let's say, quote unquote, fails a particular test or passes a particular test. And if they fail a particular test and they have the condition that you're looking for, that's a true positive. Sometimes it's a false positive, meaning they failed, but they are neurotypical. A true negative means that they passed, but they are neurotypical. Or a false negative, meaning that they passed, but they do have the condition of interest. And there are 4 metrics that scientists usually talk about. Sensitivity, which is how often are children that have the disorder detected by the screen. And there's a little formula for it: true positives divided by true positives plus false negatives. Specificity, how often are children that don't have the condition, does the screen say that they are negative? And there's the formula for that. Then you have the positive predictive value. Out of all the positive findings, how many are true positives? And there's the formula. And the negative predictive value. Out of all the negative screens, how many are true negatives? And now we're going to launch into the eye tracking. And I think that eye tracking has a huge amount of potential, not just in autism, but in lots of fields. But for our purposes, I think it can reduce the age of first diagnosis because it's super fast and objective. And, clinicians train their whole lives to be able to identify children that are on the spectrum at young ages, but it takes an enormous amount of time and energy and training, whereas eye tracking really can be done by anybody pretty quickly. It also can reveal distinct biological subgroups that will hopefully get us towards precision medicine. It can also be used as a prognostic indicator, meaning that what a baby decides to look at and what their patterns of eye gaze are when they're young could help predict their future behavioral repertoires and abilities. It could be used as a guide to treatment planning and precision medicine. Again, once we understand subgroups based on eye tracking, that might help us figure out what treatment might be best for what child. And it could also be potentially used as a marker of treatment response, right? You could possibly do eye tracking before you do a treatment, then engage in some sort of service, and then do the eye tracking again and see if patterns of eye gaze have changed. It's objective, quantitative, and it doesn't rely on clinical judgment, right? The diagnosis of autism today does have objective criteria, but there's probably a scotch of subjectivity that goes into it. So this is nice and objective. And I just want to mention that whether you know it or not, eye tracking is everywhere. There's lots of research, and pretty soon it's going to become standard just in your phone and your laptop. People are using it to understand consumer behavior, like advertisers. Where is someone looking in an ad? Concussions, it's already out there. There are physicians that are assigned to different teams. Like if somebody has a concussion, they'll run out to the field with like a little eye tracking device, and they check to see if there's any brain injury. Like, for example, are the pupil sizes uneven, or are people able to track objects smoothly? That's eye tracking. Safety, truck drivers now, there are little eye tracking machines in the rearview mirror, and they check to see if their eyes are closing or if they're looking drowsy. And then reading issues, right? Eye tracking is great for this because you can check to see, is the child skipping words? Are they perseverating on a specific word? So it's really got massive potential. It's even way more than this, but I thought I would mention that people are using eye tracking for lots of different things. And eye tracking actually first kind of got introduced or popularized in the 1960s by Yarbus. And this is what an eye tracking machine looked like in the 1960s. It just had like cameras, and they even had like these suction cups that you would put on someone's eye, and then a mirror would check to see where the eyes are moving. Of course, that would be like not particularly tenable for our population at all. But Yarbus showed that you can reliably understand what someone is looking at using this apparatus. But again, there's just no way our babies are going to be using the eye tracking machine. Luckily, eye tracking today is much simpler. And what we do with eye tracking is we do a technique called pupil center corneal reflection. And I'll tell you a little bit about how eye tracking actually works in a second, but in order to understand it, just a little bit of a review. You have your iris, which is the colored part of your eye. Then you have the pupil, which is in the center of your eye. And then you have the cornea, which actually covers the iris and the pupil. And so basically the way eye trackers work is here in this image, the eye tracking machine is actually on the bottom. It looks like a computer monitor, but there's actually a little eye tracking machine underneath. And it emits infrared light beams, which are naked to the human eye. You don't see it, but it's there. And the infrared light beams like come out of the eye tracking machine, and it illuminates the eye. And it basically creates a reflection on the pupil. It illuminates the pupil, which creates a reflection on the cornea. Then there are little cameras also in the eye tracking machine that are like looking for the position and recording the position of your eyes. And basically what the algorithm does is it looks at the distance between the center of your pupil, and you probably can't see it, but like here, right here, this is the corneal reflection. So every time you move your eyes, this vector changes between the center of your pupil and this little corneal reflection that's happening, because the light is like shining in your eyes. That's it. It's actually kind of simple. And when you're reading eye tracking papers or listening to lectures like mine, let's just all get on the same page about the metrics. So people might be talking about gaze point of fixation. That's pretty much exactly like where on the screen or exactly where someone looking. It's like the XY coordinate of a fixation, of a look. And the only thing to know here that is that it depends on your eye tracking system. If you have a 60 hertz system, that means you'll be collecting 60 fixations per second. But if you have a 120 hertz system, you'll get 120 fixations per second. 300, you'll get 300 fixations per second. So your data sheets will expand depending on what kind of machine you're using. Other things people think about are time to first fixation. Like how long did it take you to look at that little girl's eyes, right? Did it take you 200 milliseconds, 400 milliseconds, 10 seconds? That can give us information about what individuals are finding interesting. Also fixation sequence, right? You can look at this image. Some people are maybe particularly methodical and you might just look at all those cute puppies in a row, but other people, maybe they it's like an English hound, or I don't know stuff about dogs, but this center guy, maybe you love him. And you looking at him, right? Eye tracking can tell you what is this person like, which is amazing because babies can't talk to us and tell us what they like and what they're interested in and how they're learning, but eye tracking can. And then when you're reading eye tracking studies, usually people will talk about percent fixations within a specific area of interest. So right here, let's say this is actually a still image from one of our joint attention paradigms. And let's say I want to understand, is the baby like looking at this actress's face, or are they looking at this spinner object or the teddy bear? So what we do is we just basically draw areas of interest or draw, little shapes around different things. And then when you export the data, you can tell the eye tracking system, okay, tell me the amount of time and the baby looks at the truck or here or there. So it's just a percent fixation within an area of interest. Then we want to look at saccades per second. Saccades are basically, if these are fixations, the blue circles, saccades are the eye movements, right? So when you move your eyes around, you're saccading. And we want to know, is the baby saccading quickly or slowly? Because that also tells us things about how the baby's analyzing their scenes. Some people are also looking at blink rate. It turns out that there are relationships between blink rate and cognitive processing states. So if you blink less, you're kind of more cognitively interested. And like, for example, if you're hanging out with friends, and they're, blinking all the time, that's a bad sign. You want them to not blink because that means they're really interested. And this is some work that's been done by Schultz and colleagues in 2011. There's way more than this. We have average fixation duration, entropy, ratio scores, heat maps, revisits. But the 6 that I list on the screen are the more common metrics. And so when I first started thinking about, I wanted to use eye tracking as a way to identify individuals with ASD at the youngest ages possible, I thought about this study. It's a really old study by Gorin, replicated later. Basically, a mom has a baby, and the babies are literally only 9 minutes old, and a scientist busts into the room with 3 different paddles. One that has a face constellation, another paddle that has a scrambled face, and another paddle that's completely blank. And literally they go like this, and then they record and see, oh, what does the baby look at? And literally within 9 minutes, babies will spend more time looking at the face than the non-face paddles. And so I was like, I think most kids with autism probably would look at the face, but there are some kids that I know that absolutely love math, repetition. They're, really visuospatially gifted kids. And so I came up with this test called the geometric preference test for autism, which doesn't so much capitalize on, maybe, children with autism may look less at these kids, but I wanted to find something mathematical and repetitive that some kids with autism might really like to look at that could distinguish kids at super young ages. And it's basically a 60-second test. It's visual only, no sound. And again, it capitalizes on trying to identify that subset of kids with the enhanced visuospatial abilities, local processing, interest in repetition, superior math. And so we draw our AOIs. Either you're looking at the side with the kids, kind of quote unquote the social AOI, or you're looking at this kind of repetitive side, the percent fixation geometric side. Those are our areas of interest for this particular eye tracking test. And here I just want to show you some data from a typically developing 14-month-old who is taking this test. Now, the red dot is the actual data. It shows you where the child is looking. They don't obviously see that when they're watching the movie. All they see is this movie. And you can see, just by watching it, what this 14-month-old is kind of finding interesting, what they're looking at. And if you see the red dot enlarged, that means they're staring. And if you see the red dot disappear, that means the baby looked away from the screen. So I deliberately picked a movie that is kind of average, where the baby looks away every once in a while. But for the most part, kids are pretty engaged. And now you see a big fixation and a big staring. And here's a toddler who received a diagnosis of ASD at 15 months. Provisional. I pretty much just got into services. We don't really give the label until it's slightly older. But see if you can tell the difference, right? You don't even need fancy metrics. Okay, you can probably tell the difference. And so that is how we can identify babies who will meet diagnostic criteria for autism as young as 12 months. And the kids participate between 12 and 48 months. Here's one of the biggest, actually the biggest study in the field of autism research that has 1,863 toddlers that participated in this eye tracking test. And you can see here on their two y-axes, because it's a preferential looking paradigm, a baby can only be looking in one side or the other, and it always has to add up to 100. So let's say they're looking at the social side for 20%. That means they're looking at the geometric side for 80%. And here you can see all of the children who participate in our study stratified by their diagnostic status. So we had 725 toddlers with autism spectrum disorder, 103 that just had some features, 128 that were global developmental delay, 198 that were language delay, 162 that had other issues, 487 that were typical, and then we had 60 toddlers that were siblings of an affected individual. And you can see here, if we kind of draw a magic line at 69% fixation on geometric images, you notice that mainly it's children on the spectrum that are more interested in these geometric images than the other kids. And that, you know, if you use that as a criteria for determining diagnostic status, you have 125 true positives. Versus if you look at 1,138 kids who were not on the spectrum who participated, only 29 kids did that. And so if you crunch those numbers using the formulas I showed earlier, you get a specificity of 98%. That means 98% of the time, if you don't have autism, you're not going to fail this test. And the positive predictive value is 81%, which means 81% of the time, if you engage in fixations greater than 69%, the chances that you truly will meet diagnostic criteria for autism is 81%. And we wanted to also look at age and race and ethnicity to see how well this particular eye tracking test performs under different conditions. And this just shows you all those metrics. And it's very similar. There's a slight dip at 12 to 15 months, but by 16 months, everything becomes a little bit more stable. And we didn't really find any differences by race and ethnicity. So pretty much any child can watch it and it works about the same. But you do notice that sensitivity is only 17%. So that means that only 17% of the kids that participate in the GeoPref test, quote unquote, failed, right? All these kids with autism, that's one thing that I love about eye tracking is, yes, it identifies kids who are visually fixating in unusual ways, but it also identifies kids that are doing the same as everybody else, right? All these kids down here, they only looked at geometric shapes, 10, 20%. They looked at the social images, 80, 90%, just like the typical kids, right? So if we were to start making predictions about prognosis, we would say these kids on the spectrum are actually probably going to have a strong prognosis, and these kids might need more intervention because their visual attention may not be in the right place at the right time to support learning. And so what we did was we looked at our data and we stratified it based on kids that we call geo-responders, the kids that love those geometric shapes, kids that were kind of in the middle, and then kids that were more social responders that really liked the social images. We wanted to see if there really is something about experience-dependent mechanisms, meaning that if we can say eye tracking is a proxy for what they might do in the real world, you might say, okay, the kids that like the geometric shapes are probably going to be looking at other things, not so much social images, versus the kids who do look at the social images on eye tracking probably look at social stuff more in their real lives. And this could probably impact their scores, their symptom severity scores, their language scores, etc. And we also checked to see, remember we talked about saccade patterns, and we stratified the groups based on how much they were moving their eyes, and we stratified it based on when they were looking at the geometric side or the social side. Kids with autism that like the geometric images are shown here in a red dot. And you can see that when they are looking at the social images, they actually had a lot of saccades. So they move their eyes a lot. But when they were looking at their preferred type, which are the geometric images, they actually had more staring and way fewer saccades than everybody else, than all the kids in these other groups. So there is something unique, we think, about this subtype of child. And we wanted to, again, check for relationships between eye tracking and phenotype. And here, this is the Autism Diagnostic Observation Schedule. It basically helps psychologists provide a diagnosis. And on this particular test, a higher score means more symptom severity, versus on most standardized tests, like an IQ test or a language test, a higher score means more ability. So the average for those kinds of tests is usually 100. But here, this is ADAS total score. And so the children with autism that like the geometric shapes had the most symptom severity, because they're here in the red bar, and they had the lowest scales of early learning, like the overall developmental quotient, which is essentially kind of an IQ. And in the Vineland, they had the lowest adaptive behavior. So there is this relationship between performance on eye tracking and phenotype, and how well these kids are going to be doing in tests. And then we wanted to consider whether or not there was a genetic underpinning possibly for these social attention profiles. So what we did was, since we had such a large study, we just basically randomly paired 850 children that were completely unrelated, just to see if by chance there would be a correlation between their percent fixation on geometric between child 1 and child 2, total strangers to each other. And of course, as exemplified by the flat line, there's no relationship between what one person might do and another person might do, who are complete strangers and are not genetically related. Then we're like, okay, let's look at siblings who are related, but they're not twins. And we can see if there's a little bit of a stronger signal there, if genetics are in fact driving social attention patterns. And you can see that in this particular plot, ASD concordant individuals, although they're not twins, they both have autism. The correlation is 0.48, shown in the red line. So that's pretty strong. And then we predicted, we're like, okay, well, if that's the case, there is a genetic component to these social attention patterns. We're going to predict that twins are actually going to have the highest correlation. And we had a small sample of twins. We had 38 pairs, dizygotic and monozygotic. And you can see here the ASD concordant pairs, we only had 8, but it was 0.70. And these are the kids in the red, basically, yeah, these red colors. So you can see that we think there is some genetic underpinnings here. And we first talked about this in 2016, and Constantino had a really nice study in 2017 showing something very similar. And then we wanted to ask, because we think that eye tracking is a proxy for experience-dependent mechanisms, meaning that, how the child does in the eye tracking test could maybe impact what they're looking at in the real world. Of course, what a baby looks at and what a baby does is driving brain development, because brain development is just, really going full bore during the first years of life. So we had babies participate in our sleep MRI studies. They're really hard to do. Basically, we tell the parents, okay, do not let your baby take a nap. Get them super tired. Let's go down to the brain scanner, and we put them in the brain scanner, and we basically pump in little stories of different emotional valence, like it's time for bed, little sheep, little sheep. And then we check to see what, how their brain is reacting. Sometimes we just measure with no sound at all, kind of resting state networks. And that's actually what we did in this first study. We looked at the relationship between the default mode resting state network. Basically, resting state network is essentially areas of the brain that fire together during rest. And there's one network that is thought to be kind of more responsible or thinking about self-reflection, more emotional processing. And interestingly, when we looked at the correlation between the activity in the default mode network and just a basic visual network, we found differences based on eye tracking. So kids who like the geometric shapes, they had the lowest levels of connectivity between this kind of more social network and this more basic visual network than all the other kids that participated in the study. You can see the ASD kids who like the geometric shapes are in red. And the kids with autism who like the social images, who are more neurotypical, they had much higher percent signal change in the brain and connectivity. Actually, it wasn't percent signal change, it was just connectivity. And you can see that that was statistically significantly different. And then if you look at, differences between the kids who like the geo shapes and the typical, there was a big difference there as well. So we do think that this is a unique biological subtype, because we're seeing differences in eye tracking, social behavior, different standardized tests, as well as brain activity. And that was some work done by Michael Lombardo and our group. But as I mentioned, one eye tracking test only detects a small percentage of kids on the spectrum. And I was hoping to be able to identify lots of kids. So we developed 6 different eye tracking tests. 3 of them really tap more into visual social attention, GeoPref test, complex social, outdoor play. I'm not going to go into all of them, but just so you know, we do have 3, and there's a lot of similarities, but there are some differences to them. And then we wanted to think about developing a joint attention test, because we know that some kids with autism maybe have issues with social attention, but some other kids, maybe more of their issue is shifting attention, right? And we know that some kids with autism have like cerebellar hypoplasia, and there are brain regions that support shifting attention that are different based on brain imaging research. So we developed a joint attention paradigm to measure, can a child shift their attention around? And then we also know that there are some children with autism, even though they might pay attention socially, they have some more issues with speech. So you might call their name, they might not respond, or they don't seem to be picking up language that well. And so we developed some eye tracking tests to tap into their auditory processing systems. We call them Motherese or baby talk tests. And that's kind of looking at integrity of the superior temporal gyrus, which is a part of the brain responsible for language processing. And we know from lots of research that some children with autism have reduced attention to speech. Cool and colleagues first showed us this in 2005. They might, as I said, not respond to their name as much. We had a paper again with Michael Lombardo and Neuron in 2015 that you can see here. This is brain activation for a typical toddler in this brain area right here in temporal lobe that has a lot of activity in response to speech. And some kids with autism, if you track them for a year or two later and they have good language, if you look back at the brain scans that we collected when they were just babies, they're showing a lot of brain activation. But there are some kids, if you track them a couple of years and they're not talking, if you look backwards in time and look at their brain scans from 12 to 18 months, you're not seeing a lot of brain activation. So a brain imaging is a really interesting way to look at these subtypes. And so we wanted to ask the question, can we link up eye tracking with some brain imaging studies? And so we developed a Motherese paradigm, a baby talk paradigm, which you guys, if you have babies, which a lot of you do, you know that it's kind of like when you talk to your baby in a sing-songy voice like this.
Hi, Sweetie. How are you today? You are so cute.
And it basically is higher pitch, slower tempo, heightened positive affect, exaggerated contours, high rates of questions.
Hi, Sweetie.
And the reason moms do that, and dads, and actually people do that to their pets, they do it even when they're pregnant, is because it stimulates attention and learning. It improves language acquisition and reactivity, right? You're just orienting a more heightened attention for your baby so they can learn. And babies prefer Motherese speech to other types of speech from the first days of life. And so I wanted to try to use eye tracking to figure out attention to speech, but that's really hard because eye tracking, right, is more of a visual thing. So I was like, how can we do this? So I've decided that we should use gaze contingent eye tracking. And basically what that means is the baby controls the experiment. If they look over here, a particular sound file might happen. If they look over there, a different sound file might happen. And in that way, I can have the baby tell me, what do they like to listen to? Do they like to listen to speech, or do they not like to listen to speech and they prefer to listen to something else? But first we had to train the babies to make sure that they kind of got the concept. So we did some training with like dogs and cats. And we basically wanted to make sure that a baby had volitional shifts between the two sides. And they could tell us, we could see with their eye gaze. I mean, she's talking and saying she likes the cats, but we wanted to make sure the babies shifted up and back. And she triggered that, right? She looked at the cat video came on. She looked at the dog video came on. So babies all get trained. And then, actually, we developed this test. But in this particular test, if a baby looks here on the left side, they see a woman like, being super cute and talking with stuffed animals in this baby talk voice, Motherese type of voice. But if the baby looks over here on this side, they control the experiment. And what is going to happen is they're going to hear some traffic. So here's.
Ginger and Coco go night-night in my beddy. They love my blankie. They are so cute. They purr and purr. And then Ginger wakes up and jumps on my head.
And so now here's an individual in the spectrum.
Have you met Ginger?
And you can see the differences. Okay, so if we look at the data, it's very similar to the GeoPref test. But here, if we kind of draw the imaginary line on 30%, if you look at this woman, doing baby talk / Motherese less than 30% of the time, then that's considered a positive for this particular test. So we draw the magic line, and we have 67 individuals with ASD who, quote unquote, failed this test. Versus all these other kids in these other categories, only 4 didn't really listen to the woman. And if you look at the values for sensitivity, specificity, positive and negative predictive value, they're very similar to our other test, where we have 98% specificity and 94% positive predictive value. This is a paper we just published last year in JAMA Network Open. But now we wanted to look at brain imaging for this group of kids. So again, we did the, natural sleep brain imaging, and in the headphones, we pumped in 3 different types of speech. So we had mild social affective speech, which is basically one was just kind of reading a story in a very pleasant voice. Then we had moderate effective speech, where it was a little bit higher. Then we had the same Motherese kind of paradigms and sounds as we did in eye tracking. And here are basically results of 31 children who were not on the spectrum. They were mainly typical, but 1 or 2 had a language delay in this particular group. And you can see bilateral activation in superior temporal gyrus region, or basically this part of the brain behind your ear is processing language. And you see a lot on the right, because the right side of your brain is actually really critical for social emotional processing. And then we had ASD toddlers as a group, generally not having as much activation and not really having any activation at all to the right. But as I mentioned earlier, we want to use eye tracking as a way to identify subtypes, so we can move towards more of a precision medicine approach, understand the biological differences between kids. And so we basically used a particular approach called similarity network fusion, where you enter all different types of data, like a child's clinical data and their eye tracking and their fMRI. And then you're basically using fancy algorithms to understand like the distances between the characteristics of each of the kids on those metrics. And then you create these little similarity networks, fuse networks. And what you get out of the end are different subtypes, different clusters. The reason I mention this is because it's really important right now in autism to start to understand heterogeneity. As I showed in the beginning, we have individuals like, Temple Grandin, who are verbally fluent or getting PhDs, and they have this particular diagnostic label. And then you have other individuals who maybe are completely nonverbal and are massively struggling, and they have the same diagnostic label. And a lot of people are thinking, well, let's try to understand the heterogeneity a little bit more. Maybe there'll be some changes in diagnostic classification in the future if we can understand more of this, more profound type versus individuals that are already, have a lot of great skills that they can leverage. So we're doing a lot of subtyping in our center, and we use similarity network fusion, put all the kids in one hopper, and we got 4 clusters out. The non-ASD individuals, each child is shown as a little green circle, and ASD individuals are shown as red circles. What you can notice is that some ASD individuals, the algorithms put the ASD individuals with the typically developing kids, which is great. Again, pointing to the point that there are some individuals that may or may not actually need that diagnostic label. They have a lot of similarities with neurotypical individuals. But then we have these 2 clusters of kids with ASDs, cluster 3 and 4. And we wanted to see what are the brain differences between these clusters, what are the eye tracking differences. And so we looked at percent signal change in response to Motherese speech. And again, you can see that cluster 1 is the cluster that has neurotypical with 2 individuals on the spectrum. They had the greatest percent signal change. Versus cluster 4 is one of the autism clusters. They had very low percent signal change in the brain in response to this Motherese speech. And then we looked at socialization scores. You can see the same kind of trend. The most severe cluster was cluster 4. They had the lowest scores in this particular test. And then we looked at eye tracking, and you can see that they had the lowest levels of attention to the woman with the little stuffed animals. And they also had the lowest brain activation. So we're trying to connect the dots between what can we use with this technology to maybe understand like which babies are maybe at the highest risk of having really challenging outcomes from the earliest ages possible. And so that's what we see here. There are some big differences. And so the last paradigm I want to talk about is this joint attention paradigm, where we know that some kids with autism have issues shifting attention. So what we did is we developed a joint attention test. Because joint attention is really important. So let's say there's a little bird in the sky and a mom points to it, right? A typically developing toddler will look at the bird and look back at the mom, and it's a teachable moment. It's a learning moment. Joint attention is defined as the shared focus of 2 individuals on an object, achieved when one individual alerts the other to an object by means of eye gaze, pointing, or other verbal or nonverbal signal. And it's really been linked to a lot of, it's a driver of social competency and language. So we have a paradigm that we developed, which is a little bit over a minute in length. And an actress is basically like pointing to stuff throughout the video. And then we check to see if the baby is following her point. And I'm just going to show you a brief clip of what it looks like. And it's really fast. But if you just look at the baby's eyes, you can see this is the view with the baby with the mom. Then we're going to screen. We're going to go over to the view of the technician. So whenever somebody's doing eye tracking, the technician actually has the gaze replay. They can see where the baby is looking. And so you can see that the baby's eyes are moving and kind of what the technician is able to see.
How are you doing today, sweetie? Are you having fun? My very tiny.
Right there. Okay, so she pointed at the bow, he looked at the bow. She pointed at the box, he looked at the box. So this baby is following her points, right? And so that's all we ask. Our children on the spectrum are following the points or not. And this is a study that we're just writing up now for publication. We looked at the number of joint attention cycles. So the number of times the baby looked at the place she pointed to, face, and back at the object she pointed to for our group of toddlers with ASD. And 0, any toddler, if they didn't do it at all, they got a score of 0. And in autism, we had 47% of our toddlers not engage in any joint attention cycles at all, versus only 10% of the neurotypical toddlers. So what I like about this is not only as an early diagnostic test possibly, but people who are doing joint attention research, you normally have to videotape and it takes forever. And so it's kind of nice that we can leverage technology to kind of at least get an indirect sense of a baby's ability to engage in joint attention, because it has so many important aspects to learning. So that's some of the things that we're doing. And this is in comparison to kids with other issues that didn't engage in joint attention quite so much, but a little bit better than the individuals on the spectrum. And we looked at the relationship of joint attention cycles to what we could find out from the ADOS, which is the Autism Diagnostic Observation Schedule, where a clinician in real life points. And is there any relationship between what we're getting on our test and what is happening in the real world? So kind of a test of ecological validity of our eye tracking. And we had, of the 243 toddlers with autism that didn't engage in joint attention cycles, they had the highest or the most symptom severity on the ADOS, the joint attention presses, versus the kids with autism that actually did engage in joint attention cycles. They had better scores. So again, it keeps kind of coming back to the idea that eye tracking just has a lot of potential for a lot of different things. And we wanted to know, could this be replicated? There are a lot of people who have replicated the GeoPref test and have published in the literature, but I haven't yet published all of the tests. And so we wanted to see if an independent site could replicate all 5 tests. And so I gave all of our tests to Fred Schick at Seattle Children's Hospital, and he's currently in the process of writing up the study. I don't want to spoil it, but I'm super excited. All the 5 tests replicated. So yay on that. We also have a new grant to see, could this be useful for doctors? Because when a parent comes in for a well baby checkup, sometimes, clearly we can see that the children might be not meeting their developmental milestones, but nobody really knows, is it autism or just a language delay? And so we now have a new grant from NIMH testing the impact of our eye tracking tests on medical practice. We want to see if nurses and medical assistants can use it. Is it more accurate than just using, let's say, an MCHAT or some other questionnaire? Do pediatricians like it? Basically, the way it works is a baby, does the eye tracking. There's a score that's generated. Just basically, it's sort of like an autism score, giving you sort of a probability statistic of the probability that the child will actually meet diagnostic criteria should they go to a licensed clinical psychologist. And then the report's printed out. It looks kind of like this so that the doctor can see the baby's performance. But it helps just guide the well baby checkup exam in terms of giving you information about social attention levels. And work has been done in collaboration with my postdoc, Javad Zahiri. And this is the part that is kind of super exciting, because it's kind of bringing eye tracking full circle. How can we possibly use this information to take steps towards precision medicine and possibly improve treatment for children with autism? So we're doing a new study where we had 173 individuals on the spectrum. 24 were typically developing and 30 didn't have autism. They just had a non-ASD delay. And again, we need to start figuring out this heterogeneity. And so what we did was we looked at individuals with autism who participated in all 5 of the tests, and we tried to say kind of what kind of subtypes can we find across all of the tests. And right here on the left, this just shows you a subsample of kids with autism that took all 5 of the tests, and they're listed here on this x-axis. And this is percent fixation on social images. And you can see each child is a single line. So this child got 35% fixation on social images in this test and went up to 48%, but then they went down to 32% and so on and so forth. And you can kind of look at performance of each individual child. And if you find that a child looked at social images less than 50% across every single test that you gave them, we can say that they have low social attention based at least on this eye tracking. And only a small percentage of children fall into this really more severe category. 11% had less than 50% fixation. Then we find some kids with autism, around 23% of kids with autism perform great. They're looking at social stuff. They have more typical eye gaze, and around 23% of kids with autism do that. But now we have around the majority of individuals with autism, you're probably not surprised to see, really have really up and down performance. They might be really low, but then they one test they're high on, and then they're low again. And so this group right here, I think this mixed group, we can actually have some treatments that are tailored to try to boost social attention that might improve some of their social and language scores and abilities. These kids over here that are consistently really having challenges, I think are going to be the hardest to treat. They might need way more intervention and a different type of intervention. And these children up here, you know, maybe we should think more about academic and just more social support in schools, and maybe they don't need as much of the traditional kind of ABA or interventions that are generally out there. And so we're starting to explore looking at baseline profiles, not just of one test, because one test maybe isn't going to tell you that much, but across lots of different eye tracking tests to see kind of what is this child's profile, and can we kind of map onto some better treatments? We're just starting this work, so I don't have the full answer yet, but this is what we're thinking, and we have a proposal under review for this. And again, you know, we're always looking for relationships with symptom severity, and the kids in each of these three groups, you'd predict that the children who perform the worst on all the social attention tests will have the most symptom severity, and these are the guys shown in blue, and you can see that that is in fact true across all these different tests that we looked at. And then I also wanted to just quickly think about, can eye tracking be a useful marker of treatment response? And there's really not a lot out there. It's a totally new field, a totally new idea. There's a paper by Bradshaw and colleagues that recently came out, very small, just like a pilot study of only 12 children per group. They looked at eye tracking before and after an intervention, and they looked at social attention. They looked at changes in the eye-mouth ratio. They didn't find a huge amount as it relates to changes in attention, but they did find a decline in mouth looking following treatment, which I thought was really interesting. But again, it was a small sample size, and you can't look at subgroups with only 12, but it was a great first start. I loved that study. There is another study that looked at single dose randomized control. They basically looked at vasopressin, which is a hormone that's supposed to stimulate social behavior. And this particular study by Umbrecht found that eye tracking results showed that administration of vasopressin was associated with a large and statistically significant increase in attention to biological motion after the treatment. So people are starting to kind of use eye tracking before and after interventions. And then for the last just minute or two, I wanted to try to bring full circle. I didn't talk about the research of the larger center, but we are, because, you know, social challenges are at the heart of autism. It's why individuals with autism probably have a hard time getting jobs. There's high rates of suicide ideation and depression and a whole bunch of things. And so it's really important to understand where's this coming from, what's going on. So I tried to connect the dots with all of the other more molecular and basic research that we do at our center to see kind of what we're thinking, what our theories are right now. And here's the short story. Autism begins in prenatal life. There's a lot more evidence than the one thing I showed you, but let's just assume that this is the premise. And then because we know autism is genetic, right? The concordance rate for monozygotic twins, for identical twins, is super high. It could be anywhere from 0.7 to 0.9, right? So there's something genetic going on. And our research suggests there's overactivity in gene pathways that relate to cell cycle and cell proliferation, right? Basically, genes that are telling, the brain that's being made, make more cells, there's too many brain cells. There's just overactivity in these networks relate to cell proliferation. So you've got too many brain cells, and these excessive brain cells causes challenges of postnatal sculpting. I mean, sometimes, bigger isn't better. Too much isn't better because you're just having potentially a lot of noise or a lot of connections that are being made, but the environment isn't catching up fast enough to sculpt it. Because typically, for all of us, right, you're born with all the neurons you're ever going to have, but then you have synapses and connections that are made for the first 3 years of life, and then through experience-dependent mechanisms, those synapses are pruned away, and you're left with connections that are going to help you talk and socialize and do a bunch of everything. But if you've got so many cells and so many connections, that's going to make things pretty hard. And then if that is the case, that we think that this excessive number of brain cells and brain enlargement is the issue, there should be visible brain phenotype relationships. So kids with more enlargement should have more challenges. And this is kind of where the ASD phenotype is coming in. Just super fast, a little bit of evidence about that. Why do we think that there's overactivity in gene pathways related to cell cycle and proliferation? This was a study published by Vahid Gastati in Nature Neuroscience a couple of years ago. We basically looked at the network activity for these pathways, the PI3K, the RAS, and the Wnt. It's basically different gene pathways that are related to cell cycle, and we found increased levels in autism, but these were in toddlers in blood samples. And so we were like, okay, what does that mean? So what we did was we collaborated with the Allen Institute, and we said, okay, let's think about when are these genes normally expressed? So looking at a database that looked at gene expression in postmortem tissue in all these different brain regions, frontal, temporal, parietal. And over here on this x-axis, this says, 8 to 16 weeks post-conception, 12 to 19 weeks post-conception. And this tells you network activity levels. What you can kind of notice is that all of these PI3K, RAS, et cetera, they're really expressed right during the first trimester, early in development. So this is inferential, but we're thinking, okay, this is why we think that genes related to cell cycle, et cetera, are really important in autism, because they're very expressed and they're expressed early in life. Too many brain cells. I already showed you that one paper by Eric Crochane that looked, had people blind, just counting the cells, and we found too many, but it's also in MRI, right? There's tons of studies. Our lab, Eric and colleagues in 2001 published a paper called Unusual Brain Growth Patterns in Early Life in Patients with Autistic Disorder. Persigo's group in 2015 looked at 3,085 subjects using structural MRI and found that there is brain enlargement in autism based on a summary of all these studies. Again, this is the postmortem evidence that I showed you at the beginning of the lecture. And our newest study is looking at brain cortical organoids. Basically, you can grow little mini models of the brain. And here in ASD, these organoids are 39% bigger than neurotypical individuals. This is a new paper that just came out by Eric Crochane and colleagues and Alison Moultrie just a couple of days ago in Molecular Autism. You can check it out. And then the fourth part is, okay, you've got all these brain cells, and we think it's going to cause issues with sculpting. This is a paper we published in Biological Psychiatry looking at white matter tracks. White matter tracks are totally enlarged in autism. All of these tracks that are connecting brain regions that are colored are enlarged in autism. And this is just in toddlers. So again, this idea that there's maybe challenges with optimizing pruning could be real, because we're seeing this with DTI, and there's tracks that are important for language, like the inferior longitudinal fasciculus, that are particularly enlarged. And then finally, the last point is that there's visible brain phenotype relationships. This is, again, Eric's paper, Eric Crochane with BCOs. And here, these are kids with autism that had milder enlargement versus more severe enlargement. And we looked at IQ scores and eye tracking and saccades. And again, the kids that were more severe are the purple ones. And you can see, for me, because I love the eye tracking data, they had the lowest percent fixation on social images, so the most impaired social attention. And we look at correlations with like ADAS scores. Again, the ADAS is a symptom severity test given by psychologists. We're seeing correlations. So that's kind of the whole story of putting our whole lab together in a super fast way. Just to conclude, I think eye tracking has a huge amount of potential. I think it can help reduce the age of first diagnosis. It can reveal distinct biological subgroups. I think we can use it for prognosis. I think we can use it to help guide some treatments, and maybe even as a treatment, a marker of treatment response. And so with that, thank you so much for everybody who listened, and all of our funding agencies, NIMH, NIDCD, Simons, National Foundation for Autism Research, CDC, all of our great team, really, particularly Charlene Andreessen, who has done everything for me with eye tracking. And thank you, everybody. Whoops.
Thank you so much, Dr. Pierce. Maybe we can squeeze in like 1 or 2 questions. We have just a couple minutes here. So I'm just going to ask a couple that came in that are probably the most applicable to our audience that's on the call today. So first, when parents bring their babies in and they, participate in the eye tracking, what sort of report do they receive following the test? And is that something they can use, for early intervention or anything like that?
That is actually an excellent question. So when a baby comes to our center, we do a standard diagnostic workup, and they receive a report with all the standardized tests. And we just now created that little piece of paper that I showed you on the screen that summarizes the baby's performance. And so we literally just a week or two ago, maybe a month ago, started giving that to families. And I'm not sure how they're using it yet, but I do think that if you have a child that has more severe social attention issues, that is probably something that their treatment provider should be made aware of. And maybe they can work together to have more socially intensive interventions, like let's do face painting and let's, have a lot of interactions with other little kids as part of the treatment protocol versus a kid who does super well. Maybe you might need a little bit more academic. So it's just in the early days of figuring out how we can actually use this to help. But yes, we are starting to give it out, and stay tuned to see how that ends up helping intervention protocols. But yeah, they can totally give it to their treatment provider.
Great. And then how are you following these kids over time, if at all? Do they come back in for further testing? Do you just sort of follow up with the parents?
Yeah, that's a great question. So we ask families to come back in every 9 to 12 months until they each reach age 3. So if a baby comes in, let's say 12 months, we will usually see them back at 24 months and 36 months. And we provide a diagnostic workup every single time. And whenever we do our research studies, it's whatever the last diagnosis is what we'll use to determine diagnostic status. We had a paper in JAMA Pediatrics recently showing that the diagnosis of autism is extremely stable. If you end up meeting diagnostic criteria, chances are extremely high that you will always meet it. But what we did learn, which is super interesting, based on kids who keep coming back, sometimes 24, actually a big percent of the time, 24% of the time, a child comes in and they might only be meeting criteria for language disorder or global developmental delay at 18 months or 20 months, and then they come back at 36 months and they do meet criteria for autism. Point being that just because your child maybe isn't fully meeting diagnostic criteria when they're young, they still might when they're older. So there's, 24% of the kids, quote unquote, fall onto the spectrum, whereas very few fall off. So this diagnosis is quite stable, but kids, over time start to jump on and meet criteria.
Great. And then just one last question that I thought was actually a really interesting question that came in. Are you aware of, or is it known if autistic children that come in to do this eye tracking test go on to end up getting eyeglasses prescriptions? Like, does their actual eyesight factor into this in any way?
That's a really great question, because if somebody has a lot of eye issues, we do ask parents, but because they're toddlers, most of the time they say no, but occasionally they do. We don't really know. We do have a new grant to follow kids into school age. And there are some kids that we got eye tracking at young, and so we can kind of check to see. But that's an excellent question. I don't think anybody really knows the answer to that. So thank you. I'll look into that.